Welcome to Planetterrian Daily, episode 115 for July 9th, 2026. Let's get into today's research and health developments. A silicon chip uses electricity and enzymes to write dozens of DNA sequences at once. Harvard researchers built a chip that assembles dozens of DNA strands simultaneously with electric fields and enzymatic reactions. The method replaces organic solvents used in conventional synthesis with a water-based process. It produces clean output suitable for portable devices and potential data storage applications. Current output remains limited by chemistry scale-up needs. The team plans refinements to increase sequence length and throughput. From new tools for reading and writing biology, we turn to a device that measures one of the brain's most important overnight processes. A new device published in Science Advances measures glymphatic clearance through brain water dynamics rather than motion alone. Current sleep trackers rely on movement algorithms that miss fluid exchange during rest. The patch offers a direct window into clearance processes tied to waste removal overnight. Researchers see it as a step toward objective markers of restorative sleep. Next steps include validation against established imaging methods in larger cohorts. While this device watches fluid movement in real time, another team has found a way to map chemical differences between individual cells that bulk methods have long averaged out. University of Osaka scientists developed a stable probe that captures lipid profiles cell by cell within intact tissue. The approach reveals chemical differences between neighboring cells that bulk methods average out. It targets lipids associated with Alzheimer's pathology. The technique maintains precision across repeated measurements. Application to human brain samples is the planned next phase. From molecular detail inside the brain, we shift to a much older question about when directional behavior first appeared in animals. Researchers examined Sprigina floundersea specimens from the Ediacaran period. The fossils display consistent asymmetry that may represent the earliest known lateral bias in any animal lineage. The finding pushes evidence of behavioral handedness back more than 550 million years. No prior Ediacaran organism had shown comparable directional preference. Further specimens will test whether the pattern holds across the species. Evidence of ancient behavioral patterns reminds us that some biological systems still face very modern pressures. A Tasmanian Institute of Agriculture assessment modeled impacts if the parasite reaches the state. Managed hives can receive treatment, but feral colonies lack protection and could collapse. Loss of unmanaged pollinators would reduce services for several crops. Monitoring programs are expanding at entry points. Economic modeling of yield effects is underway. Engineered silica nanoparticles selectively entered prostate cancer cells and activated self-destruction pathways in mouse models. In those models, the treatment also boosted immune recognition of the tumors. When paired with checkpoint inhibitors, multiple animals achieved complete remission. The particles exploit cancer-specific surface features for targeting. Follow-up work will examine safety margins and dosing schedules in additional models. The inner ear's immune privilege status allowed sequential bilateral dosing without strong immune interference in a congenital deafness trial. Patients with one form of inherited deafness received the therapy in both ears across separate procedures. Early data show preserved safety and measurable hearing gains. The study adds to evidence that the approach can be repeated. Longer-term auditory and vestibular monitoring continues. The 2024 revision of the McDonald criteria incorporates new biomarkers to shorten time to confirmed multiple sclerosis diagnosis. Earlier identification expands eligibility for disease-modifying therapies. Realizing the criteria's potential requires refined imaging and fluid markers for accurate stratification. Clinical trials will need updated enrollment standards. Biomarker panels are under active evaluation. Regulatory agencies increasingly accept real-world data collected outside traditional randomized studies for trial emulation. These datasets can support approvals and even run prospective analyses in real time. The shift raises questions about endpoint definitions and control populations. Guidance documents are being revised to standardize methods. Several ongoing programs test the framework on existing registries. A review surveys epigenetic, proteomic, and metabolomic clocks now in use across health and disease. It outlines how clock readouts track disease risk and Healthspan trajectories. Validation against longitudinal cohorts remains a priority. Integration with clinical decision tools is discussed as a future direction. The authors note gaps in clock performance across ancestries. LDSC regression applied to meta-analyzed Alzheimer's datasets underestimated heritability compared with individual-level data. Imputed variance with high info scores also lowered estimates relative to array genotype markers alone. The study compared results across multiple LD reference panels. Ancestry mismatch between studies contributed to the downward bias. Authors recommend restricting analyses to directly genotyped variants when possible. University of Barcelona researchers used Spain's 24 election surprise to isolate shifts in economic outlook and fertility. Improved expectations correlated with higher birth rates and marriage numbers in the following period. Abortion rates moved in the opposite direction. The design separated economic sentiment from policy changes. Similar natural experiments are proposed for other settings. The PALIS tool combines homology detection with deep learning to resolve fragmented viral sequences from metagenomes. It outperforms prior methods on complex community samples. Accurate phage genomes support studies of microbial ecology and potential therapeutics. Benchmarking used both simulated and real metagenomic datasets. Release of the software is planned for wider testing. Digital twin models range from virtual patients to simulated organs on chips for disease forecasting. Validation standards and regulatory pathways are still forming. Early applications focus on dosing predictions and toxicity screening. Cross-disciplinary teams are defining data requirements for credible simulations. Pilot projects continue in oncology and metabolic disease. Researchers captured footage of a goblin shark at previously undocumented depths and locations. The sightings extend the known range of the 125-million-year-old species. Behavior observed includes jaw protrusion mechanics not fully documented before. The data refine understanding of deep-sea predator ecology. Additional surveys are scheduled in undersampled basins. The silicon chip developed at Harvard assembles DNA strands through electric fields and water-based enzymes, bypassing solvent-heavy conventional methods. This hardware approach could support portable synthesis devices and large-scale data encoding projects once chemistry bottlenecks are addressed. It moves DNA writing from centralized facilities toward distributed, lower-waste platforms. Watch for updates on sequence length and error rates in the next prototype round. Now, here's something most people get wrong about how DNA synthesis actually works. Most people picture DNA manufacturing as a purely chemical process that requires toxic solvents and complex purification. In reality, enzymes already perform precise assembly inside every living cell using only water and nucleotide building blocks. Right now, your own cells are adding nucleotides at rates up to 1,000 per second during replication. The new silicon chip simply recreates a controlled version of that enzymatic step on a surface. One memorable detail is that conventional synthesis still generates roughly 100 times more waste solvent than the amount of DNA produced. The practical takeaway is to watch for future devices that could let researchers or clinicians print short DNA sequences at the bench rather than ordering from distant facilities. Next time, we'll be watching for longer-term auditory monitoring results from the bilateral gene therapy dosing study in congenital deafness. That covers today's science and health news. Share this with someone who's curious about the latest research. I'm Patrick in Vancouver. See you next time. And if you'd rather watch than listen, find us on YouTube at NARA Network. Links in the show notes. And before you go, this show is part of the NARA Network, a family of daily podcasts on tech, science, and markets, each one a short, sharp briefing you can finish on the commute. If today earned its place in your feed, here's your next listen. Models and Agents, your daily briefing on AI models and agents. Find every show free at naranetwork.com. The Do Positive Pod is the club for people who do something about it. Episodes and the dispatch wall at naranetwork.com/thedppod. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.